In this lecture, we're going to talk about the urinary system, which consists of two kidneys, two ureters, and a urinary bladder, and then one urethra. So two kidneys, two ureters, a bladder, and urethra make up the urinary system. Primarily, the function of the urinary system is to cleanse the blood, to remove certain things, which we'll talk about in a moment, from the blood. So we'll be spending most of our time talking about the kidneys, the main organs. The ureters are then mainly just to transport that urine produced in the kidneys from the blood to the bladder, where it's stored and then excreted through the urethra. Okay, now this slide shows the functions of the kidney. So mainly it is to remove various substances from the blood, and one of the main ones are nitrogenous wastes, which just means waste products that contain nitrogen, like ammonia, NH4+, urea, uric acid, urochrome. Remember, urochrome is an old heme breakdown product. So you remember heme from when we talked about red blood cells. So these nitrogen waste products are something that the body needs to get rid of. They come mainly from breaking down proteins and nucleic acids. But that's not the only thing you need to get rid of. While electrolytes and water are not in and of themselves waste products, excess of these are. So excess water or excess sodium or potassium or chloride ions is something that we need to get rid of. So your kidney also does that. And then hydrogen ions as well, which so the kidney has a big role in regulating pH, which remember is a measure of the hydrogen ion concentration. It's very important to maintain the pH of your blood. And then in addition to that, old drugs, hormones, toxins from the blood, these are things that also need to be removed. So removing things from the blood is a big function of the kidney. Once you remove materials from the blood, substances, then that's what we call urine. So urine is a modified plasma. But that's not the only thing your kidneys do. We will talk briefly about some other functions. Your kidneys have a big role in controlling blood pressure. So we'll talk about the renin-angiotensin II system briefly. It does produce some hormones. We're probably not going to come back to this, so let me mention it now. Erythropoietin, you might recall we studied with the endocrine system. It is made in the kidney. It targets the bone marrow, where it increases the production of erythrocytes, red blood cells. And then calcitriol has a role in regulating calcium in the blood. And then also something we talked about briefly with the when we did the digestive system, gluconeogenesis. Okay, your kidneys are able to make glucose from other organic molecules, like your liver is able to do. So those are some additional functions. In this picture, we see the general structure of the kidney. So here we see the renal artery bringing blood into the kidney to be cleansed. We're going to focus on what happens to it as it goes through the kidney, and then the blood that comes out through the renal vein is going to have a lot of the substances removed that we just talked about, nitrogenous waste, excess water, etc. And the stuff that was taken out of your blood that forms urine is going to collect here in this region called the renal sinus, and then it will drain out through the ureter to your bladder. So notice your renal artery, vein, and ureter all connect to the kidney where it indents in this region called the hilus. Okay, now to understand urine production, there's really two main topics we need to focus on to set you up to really understand urine production. One of those two is the blood supply through the kidney. The kidney, if you recall, is one of the 3 places in the body where you have a portal system, which means that the blood flows through 2 capillary beds instead of just 1. So we're going to go over that in a moment. And then the other thing is to learn the structure of the kidney tubule, the million or so little tubes that are in the kidney where the urine is produced. So let's first look at the blood supply, and then we'll look at the kidney tubule. So there's lots of blood vessels mentioned here, but let's focus on the main ones. The renal artery comes off your aorta and carries blood into your kidney to be cleaned up. Once it gets in there, it branches repeatedly. These branches in here, segmental, interlobar, arcuate, etc., those are things that you need to look at for lab. But those are just the branches of the renal artery once it gets in there. And then eventually that blood is going to go into what's called an afferent arteriole. Remember, arterioles are little arteries. And then that afferent arteriole goes into the first capillary bed in the kidney, which is called the glomerular capillaries, or the glomerulus, shown up here. So the glomerulus is capillary bed number 1 in the portal system. And we'll talk about what happens there later, but for now let's just look at the blood supply. So blood leaving the glomerular capillaries goes out through an efferent arteriole and then into the second capillary bed in the kidney called the peritubular capillaries. So, and then that goes into some small veins that you can see in this view over here, like the interlobar and arcuate and interlobar veins, and then they all converge to form the big renal vein, which carries blood out of the kidney. So the main thing I want you to focus on here are these 2 capillary beds. They are separate. They are both in the kidney. So unlike the hepatic portal system or the one between the hypothalamus and pituitary that are in actually separate locations, these are both in the kidney, but they are separated by an efferent arteriole. So this is a true portal system. And as we'll see in a moment, they have very different functions. So more on that shortly. Here we see a kidney tubule in this picture with some blood, the blood supply. So in here is the glomerulus. This arrow leading into it would be the afferent arteriole, and then leading out of it would be the efferent arteriole, and then it breaks up into a second capillary bed, the peritubular capillaries, which includes something called the vasa recta. That's part of it. Okay, and then in this picture, which looks very complicated, but just to give you a quick overview of a kidney tubule, here we see a kidney tubule inside this red circle. So here's a kidney tubule in here. Basically, you see it's like the tube, and in some places it's highly coiled. There's this hairpin loop in it. So there's different segments of the kidney tubule. And this is something that you are going to be asked to learn, the different regions. We're going to go over that in a moment. This kidney tubule is mostly in the outer part of the kidney, which is called the cortex. This part out here is the cortex. So a little bit of it goes into what's called the medulla, which is deeper to the cortex. But since most of it is in the cortex, they call it these cortical nephrons, or kidney tubules. On this next picture, where the kidney tubule is actually quite similar, except it has a longer hairpin loop here that goes way down in the medulla, they call this a juxtamedullary nephron. Okay, so you will notice those 2 terms, but basically nephrons have, they're similar in that they have the following segments to them. They have, I'm going to draw one for you, a simple picture of one. So at the beginning of the kidney tubule, you have a sort of a cup that's going to catch the what gets filtered out of the glomerular capillaries. So the glomerular capillaries are going to come into the kidney tubule and they're going to be sort of nestled inside the very beginning of the kidney tubule, called the glomerular capsule. Glomerular capsule because it's like a cup around the glomerulus, the capillaries. Okay, and it kind of catches the what gets filtered out of the blood in the glomerular capillaries. After the glomerular capsule, there's a very squiggly part of the kidney tubule where it's highly folded. They call this the proximal convoluted tubule, or PCT for short. Convoluted means wavy. Okay. So that's the proximal convoluted tubule. You might remember the term proximal and the terms proximal and distal from your general anatomy. Proximal and distal are usually used when talking about the limbs, or the upper and lower extremities, where proximal means closer to the point of attachment, closer to your shoulder or hip. Distal means further away from your hip or shoulder. But in this case, we're using proximal and distal a little bit differently. But here again, proximal means closer to the beginning of the kidney tubule. And then, as you might guess, there's also going to be a distal convoluted tubule. But before you get to that, there is a hairpin loop. This is the part that's different in the two tubules we just talked about. If it's really long, goes down into the medulla, it's the juxtamedullary nephron. If it's shorter, it's cortical. So that's called the loop of Henle. And they do distinguish between the descending limb and the ascending limb, because the what gets filtered out of your blood starts in this glomerular capsule, and then it starts flowing through the kidney tubule, next to the proximal convoluted tubule, down the descending loop of Henle, up the ascending loop of Henle on the other side. Then there's another convoluted segment called the distal convoluted tubule, and then that leads into what's called the collecting duct. Okay, so that's your basic kidney tubule. Glomerular capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule leading into the collecting duct. Now we're just going to call this the collecting duct, although actually they first call it the connecting tubule and so on, but we're just going to amalgamate that and call this the collecting duct. Okay, so let's look at urine formation. And again, both are going to be important: the kidney tubule itself and the blood vessels, the glomerular capillaries, which I already drew. And then if I draw on the peritubular capillaries, it's going to look really messy, but I'll do it for you. Once the blood leaves the glomerulus and goes into the efferent arteriole, it then breaks up into the proximal peritubular capillaries that surrounded the rest of the kidney tubule. Okay, so my recommendation is to start with the simple overview of urine formation, and there are three main steps that have to occur. And the first one is called filtration. This is where this occurs, rather, is in the glomerular capillaries. So this is the function of the glomerulus. Glomerulus is another name for glomerular capillaries. So capillary bed number one, this is its function, filtration. So what's happening is as the blood enters this capillary bed, nestled in the glomerular capsule, there's a fairly high pressure in there, and it pushes the some of the water in the plasma and small solutes out of the capillary and into the kidney tubule, into the glomerular capsule of the kidney tubule. And once it leaves the blood, then we don't call it blood anymore. We call it, but we don't call it urine yet either, because urine is a waste product. It's ready to be excreted, and this is not. So what they call it is, they call it the plasma filtrate. Okay, so filtration is where you use pressure to push something through a semi-permeable membrane. Like when you make coffee, you the grounds in the coffee filter, and then you pour hot water over it, and then what comes through? Well, not the big grounds in the coffee, but the smaller molecules in there come through get filtered out. So filtration is step one of urine production. Now, you would not want to excrete this plasma filtrate because it's got way too many things in it that you want to keep. It's got lots of water in it that you need to actually hold on to. It's got lots of electrolytes in it, and many nutrients get filtered out initially, like glucose and amino acids and little vitamins, and there's lots of electrolytes in there that you need to keep. So, this is just step one. Now, if we were able to filter out only what we want to get rid of and not anything we want to keep, then that would be it. You'd be done. And that would be urine. You'd excrete it. But it's more complicated than that, because really filtration is what gets filtered out is based on size. If you're small enough to get through what they call the filtration membrane, which we'll look at in a moment, then you're going to be in the plasma filtrate. So that is why we need these other two steps, reabsorption and secretion. Reabsorption and secretion are going to modify the plasma filtrate so that by the time you get to the end of the kidney tubule, it really is urine and doesn't have things in it you want to keep. So let's start with reabsorption. Reabsorption means moving things out of the kidney tubule lumen back into your bloodstream. So you're reabsorbing them. So let me draw you a little picture here. So like here's a kidney tubule with the lumen in it. So let's say you have glucose in there. Okay, so you want to keep that glucose. So what are you going to do? You're going to move it out of the kidney tubule and into your peritubular capillaries. That's the other capillary bed that's part of the portal system. So that's called reabsorption. It's putting things back in your blood that got filtered out. Now they call it reabsorption, not just plain absorption, because it was just in your blood not too long ago. It got filtered out, now you're putting it back into your blood. Hence, you're reabsorbing it. When we talk about the digestive system, remember we just used the word absorption because in that case you're taking things into your bloodstream that were outside your body previously. So they just used the term absorption. But for this, we use the term reabsorption. Okay, so reabsorption is one of the functions of your peritubular capillaries. That's what you reabsorb things into. Water, electrolytes, amino acids, vitamins, anything you want to keep that got filtered out. And then another thing your kidney tubules can do is they can secrete things. So secretion is pretty much the opposite of reabsorption. It's moving things out of your peritubular capillaries. I'll just use an X for like something you want to get rid of, but it didn't get filtered. Because filtration is one major way we get rid of stuff in our blood. But what if it's too big to get filtered out? Because remember I said it has to be, it's based on size. So you have to be small enough to get through the filtration membrane. So what if it's too big? Or what if there just simply wasn't enough time? Because your blood's always moving through moving along, and you may not get to filter out everything you want to get rid of. So this is like a second chance here. You can move things out of your peritubular capillaries into your kidney tubule. This is the kidney tubule here. And that's called secretion. So secretion and reabsorption are the two big functions of the peritubular capillaries. Okay, and this is occurring, reabsorption secretion, in the Not the glomerular capsule, that's where filtration is happening, but the rest of the kidney tubule. The proximal convoluted tubule, the loop of Henle, distal convoluted tubule, and collecting duct. And as you might guess from having just studied the digestive system, different segments are going to be specialized have different specific functions. This is a highly schematic diagram that diagram that is just giving you an overview of the main functions in urine production. So here we see filtration. Again, we just talked about this. It's occurring in the glomerular capsule, the kidney, where what's getting filtered out is coming from the glomerular capillaries rather. And then once you filter things out of your blood, you're going to reabsorb things that you want to keep. So here we see moving things out of the kidney tubule lumen into your peritubular capillaries. This is just a neater picture of what I just drew for you. And then you can also, secrete things out of your peritubular capillaries into the kidney tubule. So 1, 2, 3, those are your big steps you want to focus on for urine production. By the time you get to the end of the kidney tubule, now it's urine, because you've taken out what you want to keep and put in things you want to get rid of. Okay, so let's look at filtration a little more closely here. So this is a close-up of the peritubular capillaries, which are inside, and the glomerular capsule, the beginning of the kidney tubule. Here we also see the afferent arteriole coming in, the efferent arteriole taking blood out. In between, it went through the glomerular capillaries and filtration occurred. Also notice, before we talk about the histology of it, notice that the glomerular capsule has two layers. It has an outer layer and an inner layer. The parietal layer and the visceral layer. So this is the parietal layer out here, and notice it's a simple squamous epithelium. And then on the inside of the cup, covering the glomerular capillaries, you have the visceral layer. Here's the visceral layer of the glomerular capsule. Those cells are called podocytes. They're called that because podo refers to feet, like podiatrist, and they call it that because the cells have like little feet or little extensions that come off, and they interdigitate with each other with slits in between. So those are the podocytes. They're also epithelial cells, a single layer. So histologically speaking, the glomerular capsule is simple squamous epithelium. And you should know that the visceral layer is also called podocytes. Okay, and then you already hopefully remember the histology of capillaries. Right? The glomerular capillaries are simple squamous epithelia, also called the endothelium. So I'm going to show you an even closer up view of this. Here we see the podocytes in red out here. See, there's its nucleus. And you see it's got these feet that interdigitate with the other podocytes. And then here in yellow is the glomerular capillary, the glomerular endothelial cells. And then in between the two, as you always see, remember with epithelial tissue, would be a basal lamina, or we can just call it the basement membrane in between, which is acellular. So all of this together is the filtration membrane. Remember when we talked about the respiratory membrane not too long ago in the lungs? That was a two-cell barrier. The pulmonary capillaries and the alveolar simple squamous wall. Well, here again we have a two-cell barrier. The endothelium of the glomerular capillaries and the podocytes of the glomerular capsule. Now, one difference here is that the plasma what gets filtered out of the plasma and enters the kidney tubule does not actually go through the cells, like through their membrane and cytoplasm. It goes through pores. See these little holes in the endothelial wall of the glomerular capillaries? Okay, this is a kind of capillary that is called fenestrated. It's got lots of little openings in it. So the filtrate, what's getting filtered out is going to go through these pores, and then it also goes through these slits. They're called filtration slits between the podocytes. So filtration involves the blood coming through the glomerular capillaries, the pressure in there pushing small solutes and water through the holes and slits into the glomerular capsule. So that's why I was saying that this is based on size. If you're small enough to get through the filtration membrane, then you're going to enter the plasma filtrate. Maybe it's informative to look for a moment quickly at what does not get through the filtration membrane. So like red blood cells and platelets and white blood cells do not. Nor do all the plasma proteins, the albumins, the fibrinogens, the clotting proteins, and all the other myriad of proteins in your plasma. They usually do not. Now, if you have an infection or kidney disease and your filtration membrane, the slits are larger, then you might get, for example, lots of protein in your urine, and that would be considered something abnormal when you have a urinalysis done, where they look at what's in your urine. So this is the filtration membrane, two-cell barrier between your blood and your kidney tubules. And here it is just written out. The filtration membrane. It's a two-cell barrier, 3 layers, but you notice the basement membrane or basal lamina is acellular. Okay, now once you form your plasma filtrate, as we were saying earlier, you have to reabsorb things you want to keep and secrete stuff that you didn't filter but you still want to get rid of. So this picture, don't get lost in the detail of it. Mostly I just wanted to show you this to show you the two-cell barrier for reabsorption and secretion. So just to orient you here, here's the lumen. So let's look at this glucose molecule. It's in the lumen of your kidney tubule. Okay, but you want to keep it. You're not supposed to have glucose in your urine. If you do, that's abnormal. They probably would test you for diabetes. But so normally we reabsorb all the glucose that gets filtered out of our blood. So what's the two-cell barrier for reabsorption? Well, if you're a glucose molecule, first you have to get through one of the cells that form the kidney tubule wall. And these cells are, the kidney tubule wall is a simple epithelial wall. Now, in some places it's cuboidal, simple cuboidal, as it is here. Some places it's simple columnar in parts of the collecting duct, and then in some places it's simple squamous. I am not going to be asking you what's where, but you do need to know that your entire kidney tubule is simple epithelial wall. That's all there is to it. That's why you can't see kidney tubules without a microscope. They're microscopic. And there's about 1 million in each kidney. So if you're a glucose molecule, you have to go through one of these epithelial cells in the kidney tubule wall. Then you go through some fluid, which is called interstitial fluid. And we'll talk more about that later. And then you have to get into the peritubular capillary, which again is simple squamous, the endothelium. And then you're reabsorbed. You're in the blood. The peritubular capillaries will eventually leave through the renal vein, and you'll have reabsorbed that glucose. Secretion would be the exact opposite direction. You'd move something out of your peritubular capillaries through the endothelial wall, and then in through one of the epithelial cells in the kidney tubule, forming the wall of it. Sometimes things are reabsorbed by active transport with using ATP, and sometimes by passive diffusion. So if you look down here, you see there's diffusion going on, active transport, facilitated diffusion. Okay, in this course we are not going to be going into how you reabsorb and secrete various molecules, but I would like you to know that these are the options, that we use various methods for reabsorption and secretion. Okay, so this looks like a lot here. Really too much. But it is very informative in that this slide shows the major events going on in the different segments of your kidney tubule. And you are not going to be asked about every single one in each segment, but I do want you to know kind of the major events going on in the different segments. So let's just go through those. We already covered the what's going on in the glomerular capsule. By the way, I'm backtracking, but I forgot to mention to you that a lot of times this region here, the whole thing is called the renal corpuscle. Corpuscle is an old anatomy term that means round thing. So the renal corpuscle refers to the glomerular capsule and the capillaries inside, the whole thing. So that's where filtration occurs. Okay, so now you got your plasma filtrate, no reabsorption and secretion going on there. Just it's filtration. Notice here they talk about the glomerular filtration rate. So you can measure, how much plasma is filtered out of your blood per unit time. So volume per time, they call it the glomerular filtration rate. Okay, then the filtrate enters the proximal convoluted tubule, where lots of things happen. Here's mainly what I want you to know. In your proximal convoluted tubule, you reabsorb about 65 to 70% of the water and the electrolytes that you want to keep. So right away, you start taking stuff back into your peritubular capillaries. Also, for the organic nutrients like glucose and amino acids that got reabsorbed, this is where, filtered out, this is where you reabsorb 100% of them. So organic nutrients are reabsorbed here. Okay, so lots of reabsorption going on, which makes sense. These are things you want to keep, so let's just get them back in our bloodstream as soon as possible. Notice there's also some secretion going on in the proximal convoluted tubule. If you need to adjust your pH, you can secrete hydrogen, ammonia, urea, creatinine. These are all nitrogen waste products. So there's secretion going on as well as reabsorption. Okay, then we get to the loop of Henle. In the descending limb, one of the main things, and I'm really simplifying this, I'm just picking out the main events. One of the main things going on in the descending limb is additional water reabsorption. Because humans have, especially our juxtamedullary nephrons, have really, really long loops of Henle, that allows us to make very concentrated urine. You can make urine much more concentrated than your blood if you need to conserve water. If you drink a whole bunch of water, you're going to make lots of very dilute urine. But if you need to concentrate it, having long loops of Henle is very helpful. So additional water reabsorption is the big event going on in the distal, the descending limb of the loop of Henle. And then for the ascending limb of the loop of Henle, one of the big events going on there is additional sodium reabsorption. You already reabsorbed some up in the proximal convoluted tubule, but here we're reabsorbing more. We need to keep lots of sodium in our blood. As we'll talk about in a later lecture, it's one of the main electrolytes. And if you recall, it's important to have proper sodium and potassium levels, very important, for example, for nerve impulse conduction. Okay, so as you can see, there's some other things going on, but those are the two main ones I want you to know. Additional water reabsorption in the descending limb, additional sodium reabsorption in the ascending limb of the loop of Henle. And then we get to the distal convoluted tubule, which is here, and the collecting duct. Now, for simplicity, I'm going to talk about them together. So the unique thing about the distal convoluted tubule and the collecting duct is that they are under hormonal control, and what they reabsorb is largely dependent on the body's needs and which hormones are being released at that time. So for example, if you're dehydrated, you're going to make a lot of ADH. Remember that one from the endocrine system? Antidiuretic hormone, made in your hypothalamus. Back when we studied that, you just learned that it targets your kidney. But now you can be more specific. It targets some of the cells in the distal convoluted and the collecting duct have ADH receptors, and when ADH binds to them, they reabsorb more water. So you can increase the amount of water you reabsorb in your distal convoluted tubule and collecting duct by using the hormone ADH. If you drink a whole bunch of water and you don't need to reabsorb any additional water, then you decrease your ADH production and thus reabsorb less water in these two segments. Thus you make bigger volume of a more dilute urine. Another hormone that affects the kidney, that affects the kidney is aldosterone, which helps to increase the reabsorption of even more sodium. So you already absorbed a bunch in the proximal convoluted tubule and the part of the loop of Henle, but you sometimes want to reabsorb even more. So the more aldosterone you secrete, the more sodium you're going to reabsorb. And then parathyroid hormone is the other one I wanted to mention. Parathyroid hormone helps us reabsorb calcium, more calcium. So here they are listed: aldosterone, ADH, and parathyroid hormone. So again, these are targeting the distal convoluted tubule and the collecting duct, not the other parts of the kidney tubule. And that's why I say that the last two segments are something we can regulate more, modulate more. What they do, what they're reabsorbing and secreting depends more on what hormones are being made at that time. Okay, so that's an overview of the different segments of the kidney tubule and what they're doing. By the time you get near the end of the collecting duct, we can start calling it urine, because you've reabsorbed and secreted things in and out of the kidney tubule, and now it's ready to be excreted. In this slide, I wanted to show you something about the interstitial fluid that's between the kidney tubule and all the blood vessels, which are not shown here, but that are in your kidney. So this is fluid, interstitial fluid. Here it is, interstitial fluid. If it's in the outer part of the kidney, they call it the cortex. Remember, if it's on the inner part, they call it the medulla. And the main thing I want you to notice is look at the color here, the way they have it going from lighter to darker and darker and darker blue. They're showing it this way because as you go from the cortex to the medulla, the interstitial fluid becomes more and more concentrated. There are more and more concentrated. And the three main solutes that you find more and more of to cause this, create this gradient, this concentration gradient, are sodium ions, chloride ions, and urea, which, yes, is a waste product, but it's also used in helping to form this interstitial gradient. So I want you mainly to know that there is a gradient in the fluid, that it's caused by these three solutes increasing as you go from cortex to medulla, and then just very generally the purpose of it. The reason you want a gradient out here is to facilitate the diffusion of water. Remember, water moves from areas of lower to higher solute concentration by osmosis. So if you want to reabsorb water from your kidney tubules, imagine as you're going down the descending limb of the loop of Henle, and you're reabsorbing water, the filtrate is becoming more and more and more concentrated itself. So if you want water to continue to move by osmosis out of the kidney tubule into nearby peritubular capillaries, then you need the water is not going to move by osmosis if the solute concentration of the fluid is not higher than the plasma filtrate is. So that's why you need this gradient out here in the interstitial fluid, so that it stays more and concentrated than the filtrate, which itself is becoming more concentrated as you reabsorb water. So the short explanation of it is that the gradient helps in the reabsorption of water by osmosis. Okay, a couple other things the kidney does. It has a big role in blood pressure regulation, and it does this there's some special cells called the JG cells. JG stands for, you'll see it in your book, I'm not going to write it out, but juxtaglomerular cells. Juxtaglomerular. Juxta means next to, the glomerulus. So if you remember how the kidney tubule, the distal convoluted tubule, this is the collecting duct, came close to the glomerular capsule here. Right in here, there are some specialized modified cells, the JG cells, juxtaglomerular cells. Anyway, they do various things, but we're only going to cover one of them. And one of them is that they make renin. Renin is an enzyme that these cells make. So the JG cells make renin. Okay. Now, when do they make it? Well, they make it when you need to raise your blood pressure, when your blood pressure is too low. Like, for example, if you're really dehydrated and your blood pressure starts to drop, then your JG cells are going to release renin. And as we'll see in a moment, renin is going to raise your blood pressure. Not it's directly, but it's going to lead to it. Okay, so the JG cells make renin when you need to raise your blood pressure. And what renin does is it converts this plasma protein called angiotensinogen, which is in your plasma now. It's one of the many plasma proteins. Remember those? They're mostly made in your liver. They hang out in your bloodstream. They have specific functions, like fibrinogen for blood clotting, for instance, or prothrombin for clotting. So anyway, angiotensinogen itself does not affect blood pressure, but here's what renin does. Renin acts as an enzyme, basically, to convert angiotensinogen to angiotensin 1. So it converts this protein angiotensinogen to angiotensin 1. Okay, and then, so now you've got angiotensin 1 in your blood, and then when it goes through certain tissues, like your lung tissue, for example, and some others, that contain another enzyme called ACE, angiotensin 1 converting enzyme. Angiotensin 1 converting enzyme. This enzyme found in your lungs and some other tissues converts angiotensin 1 to angiotensin 2. And that protein is a very powerful vasoconstrictor. And it helps you deal with low blood pressure. It decreases the glomerular filtration rate, so you're not going to lose as much from your blood. It enhances reabsorption of water and also ions, all of which are going to up your blood volume. Well, it's not listed here, but it is a powerful vasoconstrictor. So that should be added, vasoconstriction, which, if you remember from when we talked about blood vessels, that's a T and a. Vasoconstriction increases your blood pressure. It raises resistance and increases your blood pressure. So that's how your kidneys plays a role in increasing blood pressure by releasing renin, which really starts the whole pathway of converting angiotensinogen to angiotensin 1, and then other organ tissues convert that to angiotensin 2. One thing I want to mention on the side here, because it's not really related directly to the kidney, but since we all hear so much about the coronavirus, COVID-19, I just wanted to mention that they've found that the way this virus gets into your cells is through this enzyme, this angiotensin converting enzyme. It's in the cell membrane of many of your cells. It's a protein in the membrane, an enzyme membrane protein, and that's how the COVID-19 virus actually penetrates into your cells to infect them. So, just an interesting aside there. Alright, so we spent all that time on the kidney. We're just going to spend a few moments on the rest of the urinary system. Ureters, and please remember how to say that word, ureter, not ureter. You've got 2 ureters, one from each kidney, and basically they're just conduits for urine. They're just carrying the urine into your bladder for you to store it. So they're not modifying it. There's no reabsorption or secretion going on. They are just transporting it. And then your bladder is basically, in humans, it's to store the urine. So this is for convenience, right? You want to be able to store urine so that you can empty your bladder when it's convenient for you. Now, some animals can modify their urine when it's in their bladder. For example, frogs, if they're dehydrated, they can actually reabsorb some water from their bladders. But we cannot. Humans cannot. So once the urine's left your kidney, that's it. You're not going to be able to modify it. So you store it here in the bladder, and then when convenient, you excrete your urine through the urethra, which there's only one. And we'll look at the urethra in males versus females because it looks quite different. There is a sphincter here where the bladder opens up into the urethra. So this is a smooth muscle sphincter. It's involuntary. You do not have control over it. But fortunately, we do have an external urethral sphincter, which is voluntary. And it gives us, for the most part, control over our bladder. So, that's the rest of the urinary system. In females, just orient you. Here's the bladder. Okay, they call it urinary bladder, but just bladder is fine. And then here's the urethra here. See this tube coming down. So it's rather short in females, and then it opens out through the external urethral orifice. That just means the external opening out into the environment. So that's in females. Now compare that to males. In males, here's the bladder. And then look at the urethra. It's quite long. Oops, sorry. Oh, no. Sorry about that. Oops. Well, I'm just going to finish up like this. It's quite long. So let's follow it from the bladder. So here's where it first comes out of the bladder. It goes through this gland we'll be covering with the male reproductive system soon. This is called the prostate gland. So they call the urethra that goes through that the prostatic urethra. It's going through the prostate gland. Okay, then it goes through the floor of the pelvis, called the urogenital membrane. And so that little bit of urethra right there that goes through that is called the membranous urethra. It's not very long. And then it enters the penis in the male, and the rest of it is called the spongy urethra or penile urethra. So you see it's much longer in males. You've got the prostatic, membranous, and the penile or spongy urethra. And also, unlike in females, part of it is, well, really most of it is also used to carry sperm, but not at the same time. So there's sphincters that regulate all of that. So we'll talk more about that when we get to the reproductive system. Just going back once for a moment to the female, I wanted to mention that if you look at this picture with, here's the bladder and here's the uterus, where during pregnancy it's going to enlarge quite a bit. So you can see why in pregnant women they often have to void their bladder. Okay, well that completes the urinary system.